Thank you very much. Before to start my presentation, also to Trau Nutrition, Selco, and Old Nutreco Group, with which I'm collaborating different trial. The title of my presentation today, but at this point I will start with the timer to avoid to lose a lot of time, is impact of Fusarium producing mycotoxin on performance on dairy cow. And this is a good opportunity for me to share some my recent research that investigated this aspect, not just related to Fusarium, but as Swami told you, also related to the problem of this year that in the case study of Italy should be related to aflatoxin. Here a brief presentation at what are mycotoxin. Mycotoxin are toxin substance that are produced by molds, but I believe that majority of the people that are listening us know this aspect. Anyway, I would to use this bullet point to clarify some aspect that make difficult the research that we are doing on mycotoxin, in particular for ruminants. For example, mycotoxin, excuse me, are defined are often low molecular weight molecules. What this means that they are absorbed very fast by a superior organism. I remember that I tested this condition some years ago, and I verify as both aflatoxin B1 and its metabolite aflatoxin M1 can be detected in the blood just after 5 minutes from ingestion of contaminated milk. They are very stable. What this means that we are unable to reduce the contamination of feed, for example, by heat treatment or other kind of chemical treatment. So we have to manage these contaminated feed into diet. All these substances are secondary metabolites of the molds. What this means that molds produce mycotoxin not just for the growth or reproduction function, but to interact with other microorganisms such as bacteria, yeast, other molds, plants. So it's very difficult to understand when and where mycotoxin will be produced by molds. Swami already told us that more usual, it's more easy to find in contaminated feed more than one mycotoxin, and I will present for you the case of silage that for me is of particular importance for mycotoxin contamination. Again, just to recall concept that already Swami introduced, when animal ingested mycotoxin, we can have mycotoxicosis, and they can be acute if there is ingestion of high contaminated feed or diet for short period. In this case, it's very easy to understand that there is a relationship between mycotoxin ingestion and disease in the animal. But much more often, we are chronic intoxication. That means very low contamination into diet of this mycotoxin for a longer time. So mycotoxicosis very often causes hidden disorder in the animal, and the first effect is on dry matter intake usually. So there is a reduction in ingestion by animal, but there is also an immunosuppression. So the effect of mycotoxin sometimes can be direct, sometimes can be not direct, indirect, on productivity and fertility performance, passing from a reduction in dry matter intake and immunosuppression in the animal. The majority of you, and Swami presented before, that mycotoxin can cause severe economic losses in livestock sector, and we have to know that up to now there are characterized more than 80,000 different secondary metabolites mycotoxin into a metabolomic database. So this is a short chronicle of the scientific interest of mycotoxin. The research, the scientists that work on mycotoxin started 50, 60 years ago by studying the effect of aflatoxin on human, obviously, and animal too. The mother molecule, aflatoxin B1, B2, G1, G2, and metabolites of B1 that can be detected into milk M1. Then the interest for mycotoxin moved to mainly Fusarium mycotoxin, regulated around the world in some countries. Here I put abbreviation, but these abbreviations stay for ochratoxin, fumonisin B1 and B2, zearalenol, nivalenol, deoxynivalenol, also called vomitoxin, T2, AT2, and sometimes diacylcarol. And now the new problem, the emerging problem, is regard the emerging mycotoxins. So as Rami told us, these are mycotoxins that usually are not determined into field. We are starting to understand if there is a problem of contamination, and there is no regulation around the world. Anyway, there are evidence about an incidence on its status of human and animal of this mycotoxin. This slide just highlighted the majority of them come from Fusarium molds. Just cerimatoxicin is produced by Aspergillus. And my presentation will present you some scientific paper I recently published on all this group of mycotoxin. But I would start with the specific topic of this presentation by recalling a review that I wrote with people from my department and University of Denmark on 2015, and that regarded the mycotoxin issue in ruminants. I put in this slide two tables that we published in this review. Table 4, in which we summarize the majority, at least I hope, of paper published and that a verified effect of mycotoxin on rumen microbiota by using in vitro method, lab scale methods. And we used at that time 15 published work. On table 5, instead, we reported the effect of mycotoxin ingestion in ruminants by using scientific paper that verify this condition on field, or by carrying out scientific experimental trial in which probably the majority of the authors checked, for example, the individual parameter intake. And we summarize in this 3-4 page table 50 published work. For my point of view, this is few work. So we can use few information to understand at that time what is the real effect of mycotoxin on ruminants. Anyway, I use this slide to summarize the effect of specific mycotoxin on specific aspect of dairy cows. And as an example, the majority of mycotoxin produced by Fusarium strain interfere with the ingestion of the animal, dry matter intake, as well as feeding behavior. What is this could be related or an effect of this mycotoxin, or for example, the feed smell bad. All us know that the main effect of Fusarium toxin is related to disease in the gastrointestinal tract of the animal, gastroenterite, hemorrhagic bowel, or diarrhea. It's very visible on dairy farm, this effect, when there is an ingestion and intoxication by Fusarium mycotoxin. For zearalenone, this is very evident into monogastric species. There is a huge effect on reproduction performance. Anyway, for me, the more important information that we have to take at home from this slide is related to the milk performance, the effect of Fusarium toxin on milk performance. In this review, we obtained some information on on reduction of milk production due to ingestion of feed contaminated by Fusarium. We know at that time that incidence of mastitis can increase, but no information were available, or very few information were available on the milk quality. This is true for the main Fusarium toxin, but as you can see in the box below, the European Food Safety Authority published a dossier on 2011-2013, and this guide declared that we have very few information for some Fusarium mycotoxin, T2 and T2, in which the information come from all dated papers that were carried out on young animal and not adult animal, or we have absolutely no information on the effect of Fusarium mycotoxin on livestock, and this is the case of some emerging mycotoxin. Before to go on, I would to focus on the real or more expected effect of Fusarium mycotoxin lactating dairy cow and monogastric tube. So the main target organ of Fusarium mycotoxin is the intestine. And usually I use this wonderful picture that was published by Antonissen and collaborator in 2014, in which he summarized the effect of different Fusarium mycotoxin to intestine health. Sometimes the effects are clear, for example, or intestinal permeability, chair proliferation. Sometimes are less clear. For example, some mycotoxin can reduce the mucus production. Some other, for example, the zearalenone alone, can increase the mucus production. So there is still doubts about the real effect of this mycotoxin. So this year I decided to work again on this aspect, and we just published another review in which we collected the more recent scientific paper that were published in the last 7 years, and aim to understand the effect of Fusarium mycotoxin on ruminants, lactating dairy cow in particular. We were able to collect 21 scientific trials. They focused mainly on the deoxynivalenol, fumonisin, and zearalenone, and to explain what are the data presented in the graph. When mycotoxin with different effects were studied in the same trial, we associated the specific trial to the mycotoxin that is more present. So looking to the data presented in the data table in the graph, you can see that 80% of the scientific paper that focused on zearalenone reported an effect on reproduction performance. When this study focused on the deoxynivalenol and fumonisin, the 100% reported an effect on physiological immune state of the animal. The majority of this study reported an effect also on digestibility. So animals that are ingesting diet contaminated by deoxynivalenol or fumonisin reduce the digestibility of nutrient into gastrointestinal tract. Some of these trials report an effect also, an expected effect also on feeding behavior, dry matter intake, and correlated body weight, 30–50% of this study, and 50% of the study reported a reduction in milk production. Anyway, I would to put to your attention a new aspect that is now under investigation that regard the milk quality. Some study that we collected reported an effect of, in particular, deoxynivalenol, but also other Fusarium toxins on milk quality. So In this slide, the first paper that was published 2 years ago in collaboration with Traum Nutrition Group, in which we tested an adsorbent, a binder toxo mix that Suomi already presented, with the specific aim to reduce aflatoxin M1 into milk. The animals were fed 3 diets. A control 0, that means a diet contaminated by low level of aflatoxin. A control alpha, that means a diet that is contaminated by higher level of aflatoxin. This is the orange line. And when animal assumed this diet, you can see that they produce a milk that has a contamination, a concentration of aflatoxin M1 higher than the European limit. By adding in this diet the toxo and mix, we observe a reduction into excretion of aflatoxin B1 into milk as aflatoxin M1, and in particular the level of contamination come back below the European limit by reducing the aflatoxin M1 contamination by approximately 50%. And this is very helpful this year. Excuse me for the police. Anyway, from this trial, there was an effect also on feed efficiency. I, in this graph, compared directly the 2 contaminated diets, and you can see that when toxo and mix binder was used, there was an increase in the feed efficiency calculated as milk yield on dry matter intake of fat and energy corrected milk on dry matter intake. This effect was significant, and for me this was a very important aspect, because this means that also aflatoxin can interfere with feeding behavior or gastrointestinal digestion process in dairy cow. Surely we have to still investigate this aspect to better understand what is the real effect. This trial appeared very important, as Suomi told you, this year, because we have the hot and dry season that characterized the corn harvest season of this year caused an aflatoxin B1 contamination, not just of cornmeal, but also of high moisture corn, high moisture ear corn, but also corn silage. So we have to put a lot of attention to counteract as much as possible the problem of aflatoxin 1 into milk produced in Italy as case study. This is the second experiment we carried out together, and we presented this poster to World Mycotoxin Forum of this year in May. In this trial, we tested Toxo XXL, already a product already presented by Suomi, to verify if this product is able to reduce the adverse effect of moderate level of Fusarium mycotoxin in lactating dairy cow. When I refer to moderate level, I'm speaking about contamination lower than regulamentation, in particular for the oxynivalenol, fumonisin, and zearalenone, and then that can be easily found in commercial earth. In particular, the diet that we fed to this animal are control diet with low contamination level. You can see here the contamination done is around 300 ppb, dose of Zea is 43, and of fumonisin 100. The other two diets, highly contaminated by this Fusarium toxin, showed the contamination of 1 ppm, 1000 ppb for the oxynivalenol, approximately 200 for zearalenone, and again 1 ppm, 1000 ppb for fumonisin. The only difference between these two diets was the supplementation or not of toxic XL. And another important aspect of this trial is that the intoxication period was very long, 54 days, so approximately 2 months, 8 weeks, and we divided the 36 lactating dairy cow involved in this trial into intoxication period, spring with warm condition and summer period. As a result, we observed numeric difference into milk production. The animal that ingested mycotoxin contaminated diet without supplementation of toxic XL lost 1 liter per cow per day, and we observed also an increase in milk production in the diet in which toxic XL was supplemented, contaminated by mycotoxin and supplemented with ToxXL. I don't know if this could be related to the low contamination that we observe also in control light. Surely this is another point that we should understand. There is an effect also of feed efficiency, in particular the two-diet control at ToxXL showed a feed efficiency higher than 1.5. Instead, the mycotoxin contaminated diet without product has a feed efficiency value lower than 1.5. Come to the milk quality parameter, we did not observe important difference on fat, crude protein, lactose, casein as percentage or as amount, as well as no difference for milk urea nitrogen and somatic cell count. So these appear contradictory with respect to what we reported in the review. Anyway, in this trial, we had the opportunity to classify also other parameters that are related to the milk quality. These are the milk coagulation properties that we analyze with Formagras by creating a lactodynamography curve that characterizes each milk for its aptitude to become cheese. These parameters that usually were evaluated are the rennet coagulation time, the cold firmness rate, and the A13 parameter. Just to go very quickly, if R is lower, it's better. If K20 is lower, it's better. If A30 is higher, it's better. What means better? This milk had a good aptitude to become cheese. Again, we was unable to find a statistical difference, but there are numeric difference that suggested us that when animal ingested the contaminated diet, this is the case of MTX diet, the aptitude of this milk to become cheese is much lower than control diet or the diet in which we supplemented toxic XL. In the latter case, they have very similar data. So my opinion on this is that we was unable to find no difference on the main trait of the milk because we select animal for these parameters. So these parameters are genetically derived. Instead, we not selected, or we are starting to select our cow for these secondary milk quality parameter. So the effect of mycotoxin is much more evident on this parameter with respect to previous one. For me, this is another important area of research because we have to connect the effect of mycotoxin to the whole dairy sector. The last part of my presentation regards the contamination of forage. Suomi already told us that usually Ruminants are less susceptible than monogastric to the effect of mycotoxin because the rumen microflora and the particle of feed into rumen reticulum compartment are able to bind the mycotoxin or to deactivate, degrade the mother molecule or mycotoxin. And just because this was the question of previous Q&A section, this is true, but not for all mycotoxin. Johanna Fink-Remmers reviewed on 2008 this effect by showing in a table that some mycotoxin can be the biological activity of some other molecule can be reduced by rumen activity, but this is not true, for example, for fumonisin, that parts escape the rumen mostly unchanged, or in the case of zearalenone, the metabolization of mother molecule into metabolite alpha zearalenone worse the situation because the alpha zearalenone is 4 times more estrogenic than mother molecule. And this is another concept that was anticipated by Suomi. The diet of ruminants are much more complex because they include silage, hay, pasture, or fibrous co-product, and the mycotoxin that we can detect into this feed are much more diversified with respect to concentrate and protein-rich feed. So I would present you a paper that we recently published on Toxin Journal, in which we carried out a survey in Italy again on the quality of corn silage. We collected, I don't remember exactly, 65, 66 corn silage directly on farm, and we characterized this corn silage for a lot of mycotoxin, and we used a statistical technique that is able to cluster, to group this corn silage based on the different contamination level, both in terms of which kind of mycotoxin we detected into silage and concentration. So we grouped this corn silage into 5 clusters, and we defined this cluster. Cluster 1 has silage contaminated by low level of both Aspergillus and Penicillium produced mycotoxin. The cluster 2 has silage contaminated by low level of fumonisin and other Fusarium produced mycotoxin. And I consider this silage as safe, contaminated by low level of mycotoxin. They represent around 65–70% of the total corn silage that we collected, and this is a good news. So the quality of the corn silage in Italy is quite good. Anyway, there are other 3 groups. Cluster 3, Cluster 4, and Cluster 5, I put the number of contamination that are contaminated by high level of Aspergillus mycotoxin. I'm not referring to aflatoxin, but emerging Aspergillus mycotoxin. A lot of emerging Aspergillus mycotoxin. The Cluster 4 is contaminated by high level of Fusarium, emerging Fusarium mycotoxin, and the Cluster 5 by fumonisin and their metabolites. So the bad news is that 30% of the corn silage in Italy is contaminated by high level of this mycotoxin. To go very quick, you can see that the safe silage was contaminated concomitantly by 23–24 mycotoxin. To reach the higher value in Cluster 5, 33 mycotoxin that are concomitantly present in this silage, up to the 43 of the Cluster 3. As last paper research I would share you, when we visited this dairy farm, we collected corn silage, but also the milk in the bank, in the tank. So we analyzed the milk for a metabolomic profile by working with our omic group, and this approach, I can explain it better in Q&A section, is able to differentiate, to understand that the milk is produced by safe silage. These are the blue and green group, or by unsafe silage. This approach permitted also to understand what are the main metabolites involved in this discrimination, and they are connected to amino acid, peptides, and purine and pyrimidine. Now, the people that work on ruminant nutrition know that purine and pyrimidine are connected to microbial crude protein. So the idea, but I'm speculating about this because I have to realize data, is that if animals are fed with highly contaminated silage, the microbial crude protein could be reduced. So there is an interference between level of contamination for silage, but all these emerging mycotoxin and rumen efficiency, but also with an interference on the absorption of protein. So, excuse me for this delay. I will conclude my presentation with this take-home message. The effect of Fusarium toxin are deeply studied in animal, but we have very few information in ruminants, in particular to understand what is the real effect of this mycotoxin on feed behavior, feed digestibility, and much more important, because we have very few data, milk quality parameter. Anyway, we are starting to understand that there is a real effect on animal performance. Excuse me. As a statement, I can declare, because I showed just one of the two published papers that I did by testing this specific level, 1 ppm of the oxynivalenol of dry matter of the diet on a dry matter basis can interfere with animal performance. We need more data to define the same limit for zearalenol and fumonisin. A lot of regulated emerging Fusarium toxin can be found into silage, but in silage usually are detectable a lot of Alternaria, Aspergillus, Penicillium, and other more emerging mycotoxin. They are dangerous, they can cause a problem on animal. We don't know this for the majority of these mycotoxin, but surely we can use these metabolites as biomarker of safety of the silage, and I'm studying this aspect in a specific protocol. The last two recommendations regard people involved in animal nutritional aspect, research aspect on animal nutrition, because we have to understand this aspect by carrying out trials that can be compared to each other, and we should work together as in the collaboration with Trial Nutrition to understand this aspect on our animal. Thank you very much for your attention, and excuse me for these 2 minutes of delay.